So Chelsea, am I a co-host now that I can share screen?
You should be able to.
Okay, I'll try. Hello all. Here we go. Okay, so let me close this up. So what I want to do today, can everybody see that slide well enough? One of the things I'm trying to do is not use the full screen so that I can zoom in where necessary on subjects, which you can't do very readily if you're using the full screen for the show, although the full screen does give a bigger picture. So maybe I'll switch to that at some point. In any event, my goal for this talk, this is a reprise of a talk I gave at the Argentina and Chilean Botanical Congress a few years ago, where Idris Peralta and Rocio Diana organized a symposium on Solanaceae. And I wanted then to talk about the sort of the development of molecular phylogenetics on Solanaceae, and I ended up with sort of an overview of the groups of Solanaceae. I decided to cut that out and just cover a little more of the history this time. Since I've been involved with the history of Solanaceae molecular phylogenetics almost from the beginning, I thought that it would be an opportunity for me to not just tell you about that history, but also to relay some personal anecdotes. I'm getting to that stage in my career where I look back a whole lot more than I look forward. I say that with some trepidation because I miss the looking forward, but frankly, that's just where I'm at in my career. So let's dive in. I suspect that a lot of this is going to be new for most of you who are younger and just getting into this and have seen a phylogeny of Solanaceae that is sort of fully formed today, where people are working on the details, but it hasn't always been that way. So let's go. So I want to talk about that history. That'll be the most of the talk, and then I'll give a little bit at the end about where we are on Solanaceae classification and what the most recent results look like in that regard. Okay, I think I'm going to go to full screen. This will be easier to see if I do that. Okay, here we go. So let's see if I can get rid of that. There we go. Okay, so this is really where I would start the first sort of molecular studies of Solanaceae. And this is a paper by Tucker in the Annals of Botany. Whoops, let's go back. I didn't want that to go forward. Here we go. Published in 1969, and this is using seed protein serology. So this is where you inject proteins into from a plant into a bunny, and then you take extracts from other plants and you have them react against the blood serum proteins in the bunny to see how strong the reaction is, and that you can see up in the upper right. And it tells you in some very crude sense how similar the proteins are in each plant that you test against the one that you were testing it against, that was the initial protein against which the antibodies were made in the bunny. And what this showed was a series of comparisons in the lower right of different members of the Solanaceae. And I'll just point to one interesting result here, and it is that Schizanthus did not react with any of the others. You can see all the cross interactions between them, some of them much stronger than others, but Schizanthus didn't act with any of the others, and there was some suggestion that maybe it didn't belong in Solanaceae. Well, those of you who know the Solanaceae phylogeny now know that Schizanthus is perhaps the earliest diverging branch within the family. Therefore, it was the most distant thing that was in this comparison, and it didn't react for that reason, perhaps. Well, then the next thing that came along, I'm having trouble seeing this whole screen here. Let's see what I can do. Well, I can't figure it out right now. So this was the next section of Okay, I'm jumping out of full screen so I can see the whole screen myself now. The next approach was to use DNA itself, and this started in the early 80s with a comparison of chloroplast DNA by Jeff Palmer in the genus Lycopersicon, which then was the genus Lycopersicon. And this was done by a comparison of DNA fragments. And this is also probably predates a lot of your understanding of molecular analysis, but what was done is you first purified chloroplasts, which is a really detailed lab procedure. You then isolate the DNA from the chloroplasts, and then you cut the DNA with restriction enzymes into fragments. You then run those fragments out on a gel, and you radio label them, and then you can illuminate them and see the read the autorads that show the size of the fragments. And in this case, you can see, if you can follow my cursor, on the right-hand side, there are some bands on the right 3 lanes that are labeled 8.8 kilobases that are not found on the left bands, but the left bands have some other bands down lower on the autorad that are not in the one on the right. And this is where you can infer that there was some mutation that altered the site of a restriction fragment binding location, which were usually 6 bases long. And that sort of a mutation in that little narrow spot creates this fragment pattern that you can then compare. From that, I'm sorry, my computer is really slow today. I'm probably going to be problematic. So then you can take those fragments, and here's simply a table from this paper by Palmer and Zamir, and you can see where there are fragments that have cases where there are 2 fragments that add up to a signal fragment here. You can map them in a circular pattern that is the chloroplast, with each of the concentric circles being a different restriction enzyme map. And then you can take those differences and you can construct a tree. And this is really the first phylogenetic tree of anything done in the Solanaceae using molecular data. And I did my postdoc with Jeff Palmer in the late 1980s, and I had asked him, as somebody, he was somebody who was strictly a molecular biologist. He had no background or training in systematics. How is it that he came to produce a cladogram of molecular data from the chloroplast that is so conforms so well to theory about how to construct a phylogenetic tree? He had outgroups up here, a couple of species of Solanum. He had the in-groups. He had all of the labels on the tree. And he said, well, there was only one homoplasy in the whole data set. So he just started building the tree by putting together the things that had the most shared sites. And I asked, well, what about the outgroups? And he said, well, the centrifuge rotor held 16 samples. So Danny Zamir, who was doing this work with him, said, let's throw in a couple of Solanum, since we had only 13 or 14 Lycopersicon samples. So they had their outgroups just because there was a constraint of how the experiments were done. So this really dates to, that's now, what, 38 years ago, hard to believe. About the same time, there was a Japanese group that was looking at the tuberous Solanums using restriction enzymes. In this case, they didn't take a cladistic approach. They took the shared similarity of the restriction enzyme fragment patterns that they got and did a distance-based analysis, but it produces a tree that gave them some insight into evolutionary relationships. About the same time, Peter Martin in Australia was doing some of the very first amino acid sequencing for phylogenetic comparison. So this is a much different thing than DNA sequencing. Here he's sequencing the proteins themselves and determining in very elaborate experimental methods what each amino acid in the sequence was for the small subunit of rubisco. And he did this for a large number of different groups of plants, but one paper was focused on Solanaceae. And here is his tree, and you can see in the upper right, circle group of leptostomonums, and it looks like they're monophyletic. And there's a couple of other interesting things. For example, Anthocercis came out with Nicotiana, and that was something that was not really thought to be a relationship. Nicotiana belonged with Petunia and other things, right? And then here it was the Lycopersicum nested within Solanum also, which was something that had not yet been shown in any phylogenetic way to be the case. The Palmer and Zamir paper used Solanums as outgroups, but it was just looking at relationships among the Lycopersicum. So this was a really paper that deserved more, I think, acknowledgement than it got. His work was largely obscure. I think it was published in small journals in Australia primarily, but very interesting work. Okay, then Peter Tatenyi published this paper in the Annals of Missouri Botanic Garden. This was based on secondary compounds, and this was not DNA or protein sequences, and not even sequences at all so much as just determining what the compounds were. But there was some interesting results because he tried to determine major groups of Solanaceae based on these compounds, and he came up with a couple of groups that had not heretofore been given names, the Anthocercidoideae and the Atropoideae, as major separate groups within the Solanaceae. And then the big Solanoideae and Cestroideae, which heretofore had been the two subfamilies of Solanaceae, and now they are considered along with the Atropoideae and the Anthocercidoideae. And we know that these two groups, Anthocercidoideae and Atropoideae, are more closely related now to Solanoideae, but they're sort of at the base of that branch of the tree, and they are distinctive groups. And then here's another paper that came out in Genetics by Meager et al. In 1989, in which they were also looking at the small subunit of Rubisco, but their interest was in how this small gene family, there's typically 2 or 3 copies in most flowering plants, evolved in nature. And so they were taking examples from a variety of different species from throughout angiosperms, but it happened that they were interested in Solanaceae because in Solanaceae there is an extra copy that you don't find in other angiosperm groups. And this was also back before DNA sequencing was very easily done. I'll talk more about sequencing a little further along, but right now I'll just give this idea. And here they showed what they thought were the relationships among these species based on, oops, this isn't what I wanted to do, is it? No, let's go back here. Based on contemporary taxonomic ideas, and that was that Petunia and Nicotiana were more closely related, and Solanum and Lycopersicum were more closely related. And what they found in their results, if you look at the top here with the abbreviations for these species, is that the tobacco and the potato and tomato sequences all came out together separate from Petunia. And this was counter to the existing thoughts of what the phylogeny was with respect to Nicotiana and Petunia both being put together in the classification as close relatives. Here it shows that Nicotiana was closer to Solanum, and that was thought at the time to be a problem with their results. We'll see as we go ahead. And those of you who are familiar with the phylogeny of Solanaceae know that now that this is not a problem with their results. This was the first indication that they were correct in their results. I should note that I was very taken by this research because how do 3 copies on 3 separate chromosomes of a gene evolve to be more similar to each other in species than they are to the same copies in other species? So this is what's called concerted evolution, where paralogous copies, that is copies of genes that owe their history to duplication of a previous gene, become more similar to each other. And there's 2 explanations for this. And there was another botanist named Iran Pechersky who was involved in this research on the other side of it. Meager thought that this was some sort of chromosomal translocation that resulted in homogenization of these sequences. And Pechersky thought that it was probably selection acting on the proteins themselves that led to them having similar gene sequences. And both of them had evidence that supported what they thought. And I had a graduate student named Ryan Miller who did a very nice job on this several years ago, never published, unfortunately, that showed that indeed selection is probably what's going on. And he showed very nicely in Solanum that selection seems to be homogenizing the copies of the small subunit of the viscum. Oops. Okay. All right, well, this brings us up to sort of where I entered the picture. I started my postdoc with Jeff Palmer in 1988, and immediately after joining his lab, I headed off to Bogota for what must have been the third Solanaceae Congress in 1988. And I was just starting my work on Solanaceae, which was the subject of my postdoctoral project. And it was a great opportunity to meet a lot of people who worked on Solanaceae, to make connections that have resulted in collaborations that have led to many publications since then, and to helping me to gain entry into a taxonomic community that I had no prior connection to. And it was in the volume published from that the very first data that I put together was published, and that was, I worked up some of the chloroplast restriction mapping data for Nicotiana to go in there, and this was the tree that I came up with. It was a small data set, but it was is robust in certain things. For example, the Australian section, Suaviolentes, came out as monophyletic, which has been held up, and some of the other relationships in here also were supported by subsequent work that came out much later. This was Clarkson et al. In 2004, phylogeny of Nicotiana, but with more detail. Well, let me digress a little bit here to talk about restriction site mapping, because this is sort of a source of data that was heavily used in the 80s and 90s, and that went away as DNA sequencing replaced it. But you started by cutting up the DNA into fragments like I had mentioned before, but what made the project I did on Solanaceae so feasible was that in 1986 or 7, the complete chloroplast sequence of tobacco was published by a Japanese group. So with that knowledge in hand, we were able to design a set of restriction enzymes to cut up the fragment, the whole chloroplast genome in a series of fragments. And so what this linear map is a chloroplast genome, and each one of these numbered fragments is a bit of that gene, of that genome that was cut up by restriction enzymes. We then cloned all of those fragments and then used them as radio-labeled tags to reconstruct the chloroplast genome of something like 180 or more species of Solanaceae. So this was a lot of lab work, doing round after round after round of what's called hybridization with different labeled probes. Here's what the data looks like. You can see again that there are fragments that add up to larger fragments, and that indicates a mutation. Here's some of the linear maps that I put together for them, and you can see anything that's a solid line is something that is invariant or is variable and has some phylogenetic information, and the smaller lines are ones that are invariant, but they help you to align the maps. And this resulted in the first family-wide molecular tree of Solanaceae that Jeff and I published in 1992 in the Annals of Drury Botanic Garden. There has been a lot of work done since then on the family by myself and others, but the results that we found have basically held up remarkably well to the subsequent work. And details have been refined, and a few places that were weakly supported have become more strongly supported, and so on. But I'll use this as a little bit of an example to show what some of the early phylogenetic results look like. So first of all, we showed that the Solanoideae is monophyletic, but the Cestroideae, these are the two main subfamilies in the early classifications, was a paraphyletic grade at the base of the family. And this was made a lot of interpretations of characters in the family and so on make sense, that were everything from chromosome numbers to fruit types to chemical compounds and so on. We found that one of the most strongly supported large clades within the tree was this group that we have subsequently come, we called the X equals 12 clade. And this included all of the Solanoideae, but it also included Nicotiana and Anthocercidae, the Australian endemic Anthocercidae, which have a base chromosome number of 12 pairs. And the earlier groups had different chromosome base numbers. And this was a nice coincidence, not coincidence, it was a nice correlation that was resulted from its common ancestry. Early studies of chromosome numbers in Solanaceae used to say that X equal 12 was the base for the family, and that all of the variation in chromosome number that we now know in the Cestroideae was derived from it, and it's really the other way around. It also, let's see, another thing I pointed out here is that also the Nicotiana, this confirms that Nicotiana is closer to Petunia, which we saw in the meager RBCS data. This tribe that used to be defined on the basis of bilateral symmetry, corolla symmetry, came out in 3 different places on the tree, so it's not monophyletic. Jultomata sister to Solanum. This was a bit of a surprise to many people, and it has been robust to subsequent analyses. Lycopersicon and Siphamandra included in Solanum. People had thought that Siphamandra might be. It was certainly closely related, and this was confirmed. I have a note here that this confirmed what David Spooner had found in, or later found in 1993. And so I'm going to come back to some more studies of Solanum itself later, and I'll mention David's work then. Atropa with hyoscyame, this was a question that had been suggested, but no one had put it there. There's some very cool work by a former student of mine, Yuen, about the origin of Atropa that I'll mention a little bit later as well. So at this point, I want to take a digression away from talking about what's going on within Solanaceae to focus on where the Solanaceae fits into the bigger picture of things. And this really is a topic that in the molecular sense had to rely on DNA sequencing, because you can only take restriction site mapping so far in terms of evolutionary distance to have reliable results. And getting on beyond the boundaries of the family is difficult. So in this case, this is an interesting tree published in a paper by Jeff Palmer and a series of his collaborators and postdocs in Annals of Missouri Botanic Garden in 1988. This was based on a talk he gave at the Missouri Fall Symposium in 1987, which I happened to attend, or 86 or 87. And it was what stimulated me to want to go to work in his lab. But here was a tree based on RBCL sequences, and RBCL, as probably all of you know, was the starting point for a lot of the DNA sequencing phylogenies in plants in general. And to get a sequence for any gene prior to PCR, which was invented about the same time this paper was published, was a really difficult project. And so Jeff pulled together here all of the available RBCL sequences that had ever been published today. And that includes down at the base of the tree, some cyanobacteria, some green algae, there's Marcantia, liverwort, and then all the rest are flowering plants. But most of the flowering plants are, if you look a little closer, are cultivated plants, right? There's lettuce and sunflower, and there's spinach and a couple of legumes and a bunch of crop grasses. And in Solanaceae, tobacco, petunia, and tomato, and a couple of wild tobaccos. But this, at any event, was the first DNA evidence from Solanaceae brought together into a phylogenetic tree. It showed that Solanaceae belonged in the Asteridae, which was where it was expected to belong. An interesting result here shows spinach as sister to the Asteridae. This is the relationship that we now accept, and this group is called the superasterids. But this was took a lot of data to eventually confirm that relationship. But here it was showing up in the very first RBCL-based tree of plants. This was a distance-based analysis, nothing particularly sophisticated about the analysis. It did show, though, this relationship between Nicotiana and Solanum being closer to each other than either to Petunia. So that is now becoming a robust relationship with multiple lines of molecular evidence supporting it, but still sort of denied by traditional taxonomists in Solanaceae, who would point to that relationship to say that there's something wrong with the molecular phylogenies. I can recall at one Congress, that may have been in 1994 in Adelaide, when somebody, it was either Mike Nee or Bill Darcy, told me that this couldn't be correct because Petunia and Nicotiana were so similar to each other that it was hard to even justify separating them at the generic level, much less that they would not be together in a phylogeny like this. But out of this, I would want to acknowledge where some credit is due. Gerard Zurowski, who was a biochemist who worked on spinach, rubisco, developed RBCL primers for sequencing of spinach that were published in a paper of the RBCL sequence. Well, the sequence was published in 1981, which long predated this. But when people started becoming interested in RBCL as a source for data for phylogeny, Zurowski produced and purified the primers that you would need to amplify and sequence RBCL in large quantity and gave them freely to anyone who asked for them. And so we owe a huge debt of gratitude to Zurowski, who for facilitating so much work on RBCL sequencing from the late 80s, really up through the late 90s. For a decade, this was the primary source of data for molecular phylogenetics in plants. So how does this work? Well, to give you some perspective, for those of you whose knowledge of DNA sequencing and experience with DNA sequencing is basically next generation sequencing, this is going to seem preposterous that we had to go through this. But so here is a linear map of the RBCL gene in the chloroplast. Number 1 indicates the beginning of the start codon. 1434 at the right end indicates the end of the stop codon. And there's very conserved sequence at the beginning of the gene. And it turns out there's a very conserved region that we discovered by having access to the whole chloroplast genome in a stem loop region downstream of the 3' end of the gene that was the termination of transcription point. And so we developed primers for these two ends that we could use PCR to amplify. But there was still no good way to directly sequence PCR products. So we had to take this PCR product that we had amplified. We had to then cut it with restriction enzymes. EcoR5 up here near the beginning, about 100 bases in, and this XBA1 site about 100 bases from the end, or 75 bases from the end, something like that. And then we would clone those fragments into bacteriophage, which we would grow up in E. coli. Then we'd have to extract the phage out of the E. coli after it had been cultured for a couple of days. And then we would extract the DNA from the phage. And then we would use universal primers in the phage to sequence the region between these two restriction sites. And that's how we had to get each and every DNA RBCL sequence that was used in those early days. And man, was that a lot of work for each one. And then to get those sequences, we had to run these fragments out, dideoxy sequencing with radio-labeled fragments out on a gel and read these gels. And that was a lot of work. And probably many of you have never even seen these gels, but you start at the base where those smallest fragments, which run the fastest, and you read up C, A, G, A, C, A, T, A, T, A, T, G, A, T, A, T, so on. And this was not one of my gels. This is something I pulled off the web because I couldn't get any of my autorads, which are in my lab. But boy, this is clean compared to what a lot of what you had to read. So there was a lot of hours spent sitting over a light box, reading these gels. Well, so in 1992, in a symposium that was held in Richmond, Virginia, at what was then, it was before we called them the botany meetings, that on the phylogeny of molecular phylogeny of plants. It was really an early symposium on this. I gave a talk based on RBCL sequences, and this tree had 57 taxa in it, most of which that we had sequenced in Jeff Palmer's lab, but it included a number of sequences that others had done or that were published. And this was a huge data set, 57 sequences. How do you analyze a data set that big? I was in Bloomington, Indiana at the time. David Swafford, who is a name many of you may be familiar with because he developed one of the most popular phylogenetic analysis programs ever called PAWP, Phylogenetic Analysis Using Parsimony, invited me to come over to University of Illinois where he was. And we spent 3 days together analyzing this data set. I mean, this was such a big problematic data set at the time. And I emphasize that because today, one gene, 57 sequences, you could hardly even get that published anymore. But this was a huge effort in not only determining the sequences, but also doing the analysis. And there was a Solanaceae. Let's see, in the first paper, here we can see we had Nicotiana was the only sequence that we had of Solanaceae, and we had a bunch of other things in the asterity here that showed that it fit in this case, near the Boraginaceae and Convolvulaceae. That relationship isn't as strong as that long branch would make it seem. Obviously, these branch lengths aren't relevant to any strength of signal. The following year, in another paper also published in the Annals of Missouri Botanic Garden, there was a whole issue of the journal devoted to RBCL sequencing, and the big Chase et al. Tree that showed 500 sequences of RBCL for all of seed plants was published in that, along with a series of papers on different groups that emphasized different groups of angiosperms and seed plants where there was much more focused analyses. And I wrote a chapter on asterity with expanded sampling. And the Solanaceae now is up to 4 samples included. And it fell within a group that we called the Lamiidae. This is a name that Dr. John had used for a similar group, not exactly the same as this. But this was the first molecular evidence that there was a clade that represented this group in which Solanaceae, the Boraginales, Gentianales, and Lamiidae fell. We're up to 184 sequences in the asterity at this point. Well, now back to Solanaceae. So this stimulated the start, really, of looking at DNA sequencing as a source of data for Solanaceae. There was still data being generated in my lab and elsewhere on restriction site data, but I had an undergraduate student working in my lab at Colorado, Chelsea, that's back when I was still in Colorado, who sequenced this new gene, NDHF, another chloroplast gene, as well as RBCL, and we combined the sequences for those genes together in this phylogeny of Solanaceae that had something like 18 species or something like that. And it gave results that were consistent with the larger results that we had seen previously. One of the things I had done as I was putting this talk together was to go through and look at some of the citation numbers for these papers, because Solanaceae was one of the early families to be a source of molecular phylogenetic studies. And so a lot of these things that were being, that I've been mentioning were first introduced, like the primers for RBCL and the first PCR-based study of PCR and direct sequencing-based study of RBCL were Solanaceae and asterity. And this paper that was published in Systematic Biology looked at problems with and combining data from multiple sources as an effort to get greater resolution. And this one has been cited 680 times. Let's go back a little bit and look at some. Here's this Solanaceae, or the Asterity paper in 1993, 383 citations. This one, in which the PCR-based primers for RBCL was published, has 487 citations. So these were all papers that saw a lot of, had a lot of impact. In the Solanaceae 4 conference in Adelaide in 1994, I presented the results of the family-wide restriction site analyses and some early results of DNA sequencing. The publication of this was delayed for 5 years over issues with editors, as some of you who were involved may recall. So many other things came out in the meantime, but this was sort of the culmination of the work that I had done as a postdoc. Over 300 citations for this paper, even though it was in that Solanaceae Congress volume, which aren't as visible as many other published journals. Okay, and there's the early DNA sequencing that was published in there. So I want to show just a couple of more fine-scale studies that were done in my lab during the earlier years. This was by Eugenio Santiago, now at the University of Puerto Rico. Looked at the small clade that's now known as the Getsioides, that is endemic to the Greater Antilles. It's at the top of this tree up here. In many early classifications, this was excluded from Solanaceae because it had drupaceous fruits, which weren't found in Solanaceae, and it was just thought not to belong to Solanaceae by all of these sort of authority-based classifications that were prior to the phylogenetic ones. It shows that it clearly is in the Solanaceae, although near the base of the tree. And it belongs in a very interesting clade that includes the monotypic genus Metronychia from Brazil, a monotypic genus Soala from Madagascar, and the monotypic genus Duchiodendron also from Brazil. And so here's a very interesting clade. This group of 4 genera in the Antilles, 4 genera, but only 7 species. And these 3 other species form a very biogeographically very interesting group early in the diversification of Solanaceae. About the same time, I had a remarkable undergrad working in my group named Vicente Garcia. Vicente was a Filipino immigrant. His family was very poor, but he was an incredibly bright student. And he took my class in plant systematics because he loved flowers. His parents had a berry farm in Western Washington. And he was one of the top students in my class, and I talked to him about what his plans were. He was only a sophomore, I think, when he took the class. And he said he was going to be a doctor. And I said, geez, you really love plants. Why don't you consider being a botanist? He said, oh, no, I'm going to be a doctor. And that was at the end of the spring term. And the following fall, when school started up, he came into my office and he said, what opportunities or what options are available if I wanted to study botany? And so he decided to become a botany major. And he did this really neat study of the Anthocercidae. And David Simon and another guy at Canberra, whose name escapes me right now, sent me most of this material. I had been in Australia in 94 and 99 and had collected a few things myself and got fascinated by this endemic clade, Australian clade. And so I put Vicente to work on this. And he did a really nice job of documenting the phylogeny of this group with DNA sequences. And we published this nice paper together in 2003. Vicente then won an award that's given here at the University of Washington to an undergraduate honors student, 1 student a year, I think, by a very unusual endowment we have here at the university. It is to reward an honors student with the opportunity to travel anywhere he wants to in the world. And it cannot be for study. You can't use this award to go do study abroad or something like that. You have to take a minimum of 6 months and go travel to some place in the world and just imbue yourself in the culture. So Vicente went to Australia, spent 6 months in Australia, traveled around, met some of the scientists that had helped us out with this study. He came back, went to Berkeley to do his PhD with Brent Mishler, and dropped out ABD, all but dissertation. And I was so sad to see that he decided to move on to something else, but that's his life to live. Okay, this really confirmed the Nicotiana as the sister to Anthocercidae, but there were lots of data supporting that. And a subsequent paper in which Vicente and I joined with Clarkson, I can't think of his first name, and Mark Chase at Kew to combine their Nicotiana data with our Anthocercidae data, came out a couple of years later. Another study that came from a student in my lab, this was another interesting anecdote. I had an undergraduate exchange student from Sichuan University, came to my lab at age 18 and wanted to study plant phylogeny. And so he actually worked on a project on passion flower in my lab that never got published, although it generated a lot of great data. Some of the data was published later. But then he had to go back for his senior year to Sichuan to do an honors thesis, senior thesis. And so I contacted a colleague of mine at the Institute of Botany in Beijing and arranged for him to go work in Beijing and work on this topic study of the placement of and history of Atropa belladonna. And he did really nice work using waxy to show that there's a transposon in the middle of waxy. All of the Hyoscyae are tetraploid in origin. Atropa is hexaploid. And he showed by DNA sequencing, producing the phylogeny, and then the study of this retroposon, that the 3 copies of waxy in Atropa, 2 of them have the transposable element and 1 does not. And that implies that Atropa is of hybrid origin between a Hyoscyae ancestor and some unknown and presumably extinct diploid that gave rise to the 3rd copy of the gene that does not have the transposable element in it. This is a very nice, elegant work that was published in 2006. Well, around this time, there was some other work that was going on. So this was a work by Martins and Barkman in 2005. Helene Martins was an undergrad working with Barkman, using a gene for salicylic acid methyltransferase, or SAMT. And they took on the study of Solanaceae, and since there was some data for Solanaceae already for this gene, and I provided them with a bunch of DNAs, and they got a very nice result that, that tracked what we had found almost perfectly in our chloroplast DNA analyses. We had a monophyletic X equals 12 group. We had Anthocercidae and Nicotiana coming out together. We had the separate paraphyletic Cestroideae, Schizanthus, sister to the rest of the family. It was a really nice study. No one has ever gone on to do anything else with the SAMT in this paper, though cited 62 times. I don't think ever quite got the recognition that it deserved for its contribution in confirming a lot of what we know about phylogeny of Solanaceae using nuclear data for the first time. Okay, let's jump into Solanum a little bit, because Solanum is such a huge part of Solanaceae. And while I've sort of avoided Solanum myself, apart from some collaborative efforts over the years, in part because there are so many great botanists that work on Solanum. Sandy Lynn Bowes, David Spooner, Mike Nee formed a real great working group that got that huge grant that Sandy mentioned last week, that did so much, has done so much work and was the starting point for continuing work. But there's some really interesting early papers. So one was by David Spooner in 1993. And this was, he was working with Bob Jansen And they looked at the tuberous solanums and Lycopersicon and that related group of Solanaceae, solanum species, and really showed clearly that Lycopersicon was nested within solanum. Well, of course, Lycopersicon the tomatoes was a solanum in Linnaeus's mind, but it had become pretty entrenched in the literature that Lycopersicon was a separate genus. All this agricultural literature, and there were laws written that specified Lycopersicon as the name for tomato, and rules and regulations and governments around the world. And when they came out with this and said, we have to change the name to Solanum, it got so much bad press. So no one will accept this. It's crazy. You can't do that. This is a problem with using phylogeny as a basis for classification, and on and on and on. Well, nobody gives it a second thought today. And I and many others were sure that that would be the case, but it did cause a lot of consternation at the time. And David, many of you know David was a little nervous about this when it was published, but I thought this was a great study, an early study on Solanum molecular phylogenetics. I did include a number of solanums in that restriction site study I did as a graduate or a postdoc with Jeff Palmer, and we did publish a paper in 1997. It was the first sort of genus-wide molecular phylogenetic study. It had 36 species. We found a monophyletic Leptostomonum. Many of the sort of major clades and relationships at some of the tips have held up, although with so much more data, there was just so much missing in this tree that there's a lot that it didn't address. At the same time, in fact, the same year in the same journal, Lynn Bowes and I published our first paper together. Lynn, who of course is at the University of Utah, I don't know if Lynn is on today, I hope so, came over to Boulder, where I was, and spent a few months working with me on learning some DNA sequencing methods, and we published this paper together using DNA sequences, the first DNA sequencing study of Solanum that was in any sense broadly sampled enough to be meaningful, using the gene NDHF, which I had developed earlier that I mentioned a little bit ago. So this was published in 1997. It also had monophyletic Leptostomonum, but I think we only had 1 or 2 samples of Leptostomonum. The sampling was rather different. But this formed the basis, and really a starting point for a great career on Lynn's part, doing lots and lots of DNA sequencing. I was happy to be able to collaborate with her. She's been a great collaborator over the years. We published a few more papers together subsequent to this, but mostly she's worked independently on the subsequent work. Here is another interesting paper that came out of David Spooner's lab by Iris Peralta when she was a postdoc working with, or a PhD student working with David in the late 90s, published in 2001. And this was using the gene WAXY, or the gene for the granule bound starch synthase enzyme. And I say here that this is the first nuclear single copy gene used for Solanum phylogeny, and really for Solanaceae phylogeny of any sort. And WAXY is a big complicated gene. The map at the top of the linear map of the gene at the top shows all of the exons with introns in between, and they used a region that covers the most of the first half of this. It was first used in grasses, I think, but then Iris developed additional primers that made it useful for Solanum, and I showed you a tree earlier from my former student, Yuen Yau, who is now, I think, an associate professor at University of Connecticut and is doing very well for himself, although he's moved on from systematics. And the tree down here shows the results from her study in the tuberous solanums, which for any of you who know that group well, probably are not surprised to see a lot of, the resolution isn't great when you're using just a single gene for that group, a very complicated group for its phylogenetics. Okay, well then Lynn, going on working on her own, published a paper on in the monographs in Zurich Botanic Garden that was commemorating Bill Darcy, who had died in 1999 of prostate cancer. And I you know, that has hit home to me when I had a prostate cancer diagnosis several years ago and had surgery that successfully treated it. But, you know, prostate cancer used to be the leading cause of cancer death in men until methods for diagnosis became better. In any event, that personal aside behind us, Lynn did a extensive sampling throughout the genus, and this was really the first genus-wide extensive sampling of Solanum for any molecular data. And this was based on NDHF sequences, published in 2005. And a couple years later, with Terry Weiss, a postdoc in Lynn's lab, they added a couple more DNA regions, waxy and the TRNTF region in the chloroplast, and sort of replicated that study that Lynn published a couple years before with one gene, but now with 3 genes and with much more powerful results. And this was a real turning point in our understanding of phylogeny of Solanum. Okay, so that's Solanum. In the 2006 Congress in Madison, Wisconsin, I showed this tree, which was a family-wide, we're back to the whole family Solanaceae, of based on 2 gene regions, NDHF and the TRNLF region, with 188 species. And this became the basis for a paper that was published in 2008 that I want to talk about a little bit as the basis for the starting point for our current classification of Solanaceae. I'm just about out of time and I'm just about out of slides. So let's go here. So here is that tree from our 2008 paper, the convolvulaceae and the outgroups at the base. And this is the classification that we developed that was also published in that same volume with from the Madison Congress for the classification of Solanaceae. I've updated this a little bit to reflect some of the changes that have come recently, especially things in the physality has been where a lot of changes have occurred. The Wanaloidae, or what used to be Wanaloidae, now we call the Solandriae, has changed some. But for the most part, this structure of this classification has stayed pretty much the same. I would say, though, that I wanted to note that in this paper, between Lynn and I, we had 4 co-authors that were all students who had worked in my lab and had done theses or parts of theses on Solanaceae. Hala Abdul-Majid, who was our Egyptian botanist who worked on Hyoscyae. Eugenio did the Getsioideae work. Vicente did the Anthocyrsidae work. And Sarah Collier, who did work on the physality. Okay, so where are we now? I just want to close by showing some of the fabulous big picture phylogenetics that have been done in recent years. This was a publication by Emma Goldberg et al. In Science in 2010 with 995 taxa and 8 loci. This, I should note, has engendered 349 citations. Let me go back one and see what I can say about this. So this paper that Olmstead et al. Took taxon was over 400 citations. And then in 2013, Tina Sarkinen and Sandy led this the second big phylogenetic study of Solanaceae with 1,075 taxa and 7 loci. These are the 2 figures of the family-wide and then the genus Solanum trees. And then in Stacy Smith's lab is where some of the new work has been happening in recent years. Eng and Smith published this tree with now 1,341 taxa and 5 loci. They were interested in studying the evolution of flower color. And so they had to expand the sample of red flowers and red flower relatives in order to make the tree effective for their purposes. Did some really nice work with that. And then this Sarkinen et al. Tree has been used in another paper led from Stacy's lab with Julia Dupin et al. In Journal of Biogeography, looking at the biogeography history in Solanaceae. And from that, we can infer the origin in South America and migratory routes and the number of cases of migrations around the globe from that. I just want to end with this, that given this work that we've studied on the phylogeny of Solanaceae worldwide and its biogeography, since then, there's been some fabulous fossils found in South America by Peter Wolf and his co-workers that have implications for the dating and the history of Solanaceae. And I hope Rocio doesn't mind my saying that I recently reviewed a great new paper with another one of these physioloid fossils in it that talks does a very good job of marshaling the evidence for where to place this within the Solanaceae. So I think this will help refine these broad scale historical studies. I would say that for future work, here's a small tree I published in a paper a few years ago about the Solanaceae in the red box at the bottom, but the sister group, Convolvulaceae, has now a very well resolved phylogeny for it. And the rest of the Solanaceae, which are these small groups primarily in Africa, I proposed then that this represented a deep Gondwanan relationship with Africa, Eastern Gondwana, and South America being the three main blocks of Gondwana when it split up, being the historical ancestry of these three groups. And I think we could get at that in much greater detail with additional work, better fossil data, and so on. Okay, well, there's just too many people to thank than I could possibly name for the work that I've done in collaboration with so many others, including many of you, hopefully, who are listening today. And if I don't have you on this list, I apologize, but it's been a great career working on Solanaceae. I should note, I do have NSF support listed down here, and I've been fortunate, blessed really, to have NSF support for something like 35 years, but I never had a dime of support from NSF or anyplace else to work on Solanaceae, except for that time as a postdoc in Jeff Palmer's lab. All of this is work that I've managed to do sort of on the side with funds that, where there was enough extra funds from NSF to support a graduate student or an undergrad project or one thing or another in the lab, or through collaborative efforts. And so I love Solanaceae. It's always been sort of a sidelight to my main funded work, which has mostly been on Laniales and angiosperm, deep angiosperm phylogeny. But that's it. I've probably used all the time we have allotted, but if people want to stay and talk, I hope we can do that.
Thank you. That's really great. It was really nice to walk through the history of everything and what's going on now. It'd be nice if we maybe do 10 minutes worth of questions, just be mindful of everyone's time. So if you have questions, you can either unmute yourself and have your video and you can ask questions, or you can do it in the chat, whatever works for you.
Any questions?
I'll ask something, Dick. I'll always ask the first question. So what do you think the next steps are?
Well, that's a good question. I really am looking forward to seeing the refined dated phylogenies for the group, and hopefully that will come. And as I mentioned right at the very end, I'd like to see this expanded to all of the Solanaceae, because that's a really well-defined group. The Convolvulaceae have patterns of biogeographic history that are contrasting to the Solanaceae. They clearly are not, or did not originate in the Western Hemisphere. I think that would be interesting. Family-wide, all I can say is that I think we are doing a really good job of there's not much more we can do in the way of data. Whole genomic data, yes, would be great. And I suspect that will be forthcoming. So we'll see some resolution from that. There's probably going to be some really interesting conflict between nuclear data and chloroplast data, since most of our big picture stuff is chloroplast data now. That may help us resolve things here or there, but it will probably also create more questions that will lead to more interesting research. I don't know. What do you think, Sandy?
Well, I don't know. I think we should have a bet as to whether whole genome data is actually going to help at all. I'm not convinced. I remain to be convinced. I remain
Yeah.
to be convinced. But as you know, I'm quite open to all kinds of things. So we'll see. We'll see what happens.
I mean, there's so many things that we can do to interpret evolution. And Stacy Smith and her lab is doing a great job on things, and elsewhere as well, that using our big phylogeny. So I'd say, you know, having it well resolved, having it well dated, will lead to questions that can be addressed that we might not even be able to conceive of right now.
We do have a question in the chat. So do you think the analysis of metabolic profiles and chemotaxonomy has a role to play in the definition of phylogenetic relationship in Solanaceae in the time of next generation sequencing? And at what scale would this be useful?
So that's an interesting question. And I have a perspective on that that is based on my having gotten involved in systematics and phylogeny as a graduate student, looking at morphology and then getting into molecular biology. And I think that the same can be said of chemotaxonomy, and that is this, that while it has some interesting insights into phylogeny, I think the other way around is where the real value is. Let's use these detailed, precise molecular phylogenies and dated phylogenies that we're developing to get a better understanding of the evolution of secondary chemistry in plants, for example. Because we do know that those pathways can go back and forth, and that there's elements of homoplasy that can be interpreted, I think, in light of phylogeny. Just as we're gaining a better understanding of morphology by having molecular phylogenies that we can use to interpret morphology. So I think that's the relationship here. And that's one of the untapped areas. There have been a few taxonomic studies of chemical compounds by people looking at insects, for example, that feed on Solanaceae, and but I think there's more that can be done in that area. I see a question from Alison Caldwell about Goodspeed's work on Nicotiana, and there's many layers of hybridization in the history of genus. So I think that there's other cases besides the Atropa one that Yuen resolved. He was fortunate in finding that transposable element that really nailed it, and you can't count on finding that in every group of organisms you study. And Nicotiana is a complicated genus, and one in which I think part of the reason that there hasn't been more work done is that the people who have tried to do molecular work on Nicotiana have run into problems with getting usable data. And some of the very finest work on the problems with ITS sequencing came from somebody, I can't remember his name, who was trying to do ITS sequencing in Nicotiana and discovered that there were all these paralogs and that there were complicated getting results because they came from different ancestry. So Nicotiana is one where I think a lot of work it's potential for a lot of work. There's everybody, you know, there's good access to the different genetic material for Nicotiana. Sandy, you had suggested Stephen Dodsworth could say something about that.
Is Stephen here? Because Stephen's been doing a lot of work on molecular phylogeny. Stephen with Mark Chase and Martin Christenhuis and them have been doing lots of work with Nicotiana recently. Stephen, are you here?
Sure.
Not here, but there are good phylogenies of Nicotiana.
Yeah.
And there is a lot of alloploid hybridization, but that's pretty well resolved, and the ages of that and what happens to genes and gene conversion with age of allopolyploidy and stuff. So Nicotiana is pretty well studied.
Yeah.
Mark Chase is just embarking on a very big study of Nicotiana in Australia.
Oh, great.
Which is going to be very, very cool.
Yeah, there's some of the best early work using chromosomal painting, where you use different colored dyes to label different chromosomes, was done on Nicotiana that showed some of those polyploid ancestry or hybrid ancestry. Anna Rita asked about the time of the convulsulaceae converging to Solanaceae. Hi, Anna.
Hey.
This is a really good question. I think it's obviously older than the origin of the diversification of Solanaceae, and that now needs to be reassessed in light of these fossils, physioloid fossils. So I'm curious to see how it goes. But you know, I was struck by just looking at the Solanaceae, Convolvulaceae, and the rest of the Solanales, that there seemed to be clear-cut clades that originated in each of those in sort of the major sections of Gondwana. So that would put them in the vicinity of 100 million years old for the time that they diverged from each other. But that's just based on crude assessment and a little bit of evidence from the geological history. It really needs to be assessed in more detail.
So what do you
take that on?
Sorry, I'm sorry. Yeah, sorry. I don't know if you can hear me well. I have a bit of a distraction, but I was going to say, so I was going to make use of this opportunity to ask also people here who work on Solanaceae, what do you think is necessary so that we have better estimates? Shall we work together in terms of what's finding fossils or molecular data, or what do you think is kind of the next step forward?
Well, others may have something to chime in here, but you know, one of the questions I had on my mind here for the group, or not so much a question, but as a teaser, let's put it this way. I look around the audience here, and I see people like Sandy and Lynn, and you know, I know that Mike Nee is retired, David Spooner is basically retired. A lot of the people that I look to as senior figures in Solanaceae are long gone. People like Richard Lester, Bill Darcy, Jack Hawkes, Armando Hunzik, or David Simon. We're at, I think, another transition time, because Sandy and Lynn and I are going to be moving out of the picture before too long. And I think that there's probably people in this audience that I look forward to being the ones who will address this next. The question that you're asking is that, where do we go next? And you know, I have no crystal ball to tell me who's going to become the next people taking leadership on this. I think Stacy Smith's lab is doing some really good work, and she's been training some really good people. And there's more than one nexus in Latin America of good people working on Solanaceae. You know, Latin American institutions have been, in the past, a little hamstrung by the economics of doing some of the sorts of work that we've been talking about today. But I'm not sure that that will be the case going forward. I think there's, you know, we're much more internationalized now than we were 20 or 30 years ago in science. So,
I'll stay tuned for developments. Send me the papers. I want to read them. That's my cry out to everyone. Yeah, well, I'm really curious.
On the subject of the next generation, I did something for the 2006 Congress in Madison, Wisconsin, which was to pull together all of the photos that I could get and that I could get people to donate to create a photo gallery that we showed in that Congress of people who've worked on Solanaceae, going back, you know, as far back as I could get things and contemporarily. And so this is sitting on my computer. And there are a lot of new people, new faces, including people in the audience today, working on Solanaceae. I think it would be great if we could somewhere, somehow, someone could create an archive of images of the people who work on Solanaceae. And I'd be happy to send everything that I have to someone who could help set that up. If, I see Chelsea smiling. She might be able to work on something like this. I think Sandy once said maybe the British Museum would be interested in, you know, who knows where it will be. But it's the sort of thing that there has to be someone who's interested in the archiving.
I'll put on Solanaceae source.
Well, there you go. And hopefully someone will take that over, you know, continue to support that.
Yeah, I just got to get somebody to help me with it.
Yeah, I mean, that's the question. I mean, I look at my lab, I have freezers full of DNA, and I have no idea.
Ooh, you
you
need to archive that at the Smithsonian, Dick.
Well, that's a possibility.
They'll
My
take it.
institution has said that they're committed to preserving it, but we don't really have the facilities for it. So
The Smithsonian will take it.
okay. Well,
Oh my God.
Okay. Any other questions here? How do you think Solanaceae? Edith says, how do you think Solanaceae phylogenies can help species conservation? Well, that's a really interesting question as well. There are some issues with endemism and areas that have, this subject of phylogenetic diversity as a measure of conservation value of a given region or flora, where you can use phylogenetic information to justify conservation measures. I think that might be one answer to that question. There's some really fabulous studies done, for example, Edith, in California and Australia, and I understand now in press, one for all of North America. Well, that's, I think there's opportunity to use phylogenetic measures in a regional scale, not just so much for the individual species.
Thank you, Richard. I just want to say that it's a pleasure to me to see so many good friends. Richard, Sandy, Lynn, Helen, Aaron, and just as a reflection of this situation everywhere in the world, that even though the coronavirus is really difficult to deal with, it gives us the opportunity to meet again.
Yes, I think you're right.
I think this is a great idea, and I hope that most young people can join, because you did an exceptional presentation about the synthesis of this history of the Solanaceae studies. And when you said that the hard work that it was at the beginning, it was really hard. I mean, to clone the fragments, to produce the bacterial, and those times are gone, but now there are more opportunities. And I think that working with conservation and evolutionary studies related to the environmental factors is another step, I think. This is just my thought. But, well, my greetings to everybody. And Paul, you are there too. So thank you again.
Good to see you. There's a question from Edelina about what kind of habitat do Getsio-dendron, but I think you mean Getsioides, occur in. So the Getsia occur in scrub habitat, dry scrub habitats in the Greater Antilles. I've collected it on Puerto Rico and Cuba, 2 or 3 genera in Cuba. It's never an abundant representative in those floras. It's a very interesting group. It's related, as shown, to Duchiodendron in the Amazon, which also was not considered part of Solanaceae in early taxonomic treatments, which also has sort of a tough, leathery, husky, sort of drupaceous fruit, which may be a trait that they share. Metternich I've seen once in Brazil, although we have it growing now in our greenhouse. I don't know how many places have that in cultivation. It's a fabulous shrub with big, beautiful white flowers that is sister to the Getsioides in the Antilles.
All right, well, thank you so much. This was really informative, and I think it's like definitely a conversation that we'll keep having and thinking about, and we'll see where Solanaceae goes and what we learn about it. If you want to, you can make your screen available and wave and say bye to everyone. And next week, Stacy Smith will be giving a talk on floral color within Solanaceae, which should be really good.
All right. Thank you all.
Thanks, Nick.
See you next week.